Okay, so reminder that the first exam's next week, Thursday in here. And then there'll be a take-home version. I'll probably get that set up so that it will open like right after this lecture on Thursday and go for like 24 hours or something like that. So you can have and pick and choose when you want to do it. So I'll talk to you more about that as I firm everything up. So the content is probably going to work through what we're covering in this slide set for the exam. We'll see. I'll have a better feel for that by the end of today. Any questions about exam stuff? Yes.
Saturday, Wednesday, like take home. Is that just gonna be open till like midnight on Thursday?
I'll probably get it to a 24-hour open window. So I'll probably open it like 11 a.m. on Thursday and close like 11 or noon on Friday, something like that. Okay, so succession. Need to think a little bit about that. You'll often hear this word niche. And when microbial ecologists think about it, this is kind of how they define it. Each population has a specialized functional role, and that's their niche in that ecosystem. And we're going to see how when microbes come into an ecosystem, just by their presence and their activities, they can alter it in such a way that allows for succession to occur, where then other microbes can get in, whereas before they couldn't until that particular microbe came in and did its functions. So, you know, it's kind of the idea of thinking about the habitat, thinking about the selective pressure of that habitat, and then microbes that are there, microbes that are coming in and leaving, and that you will have some populations that are not going to hang in there because they're not as well adapted. And so they'll get phased out and others will come in to replace them. Depends on the ecosystem, how complex this will be. So this can get pretty complicated, especially in the microbial realm. And you've heard these terms in macroecology classes, the idea that you have pioneers, they're the first to come in to a habitat and start setting up camp and living their life. And so that's the so-called primary succession. So these are typically microbes that would be good at dispersing. Maybe they have the ability to move with flagella, or there are other ways to move besides just flagella. They can glide on a surface, for example. So there's a couple of different strategies there, or they could just be floating along in the water current that takes them to a new habitat. So that's another option. And so as they are in that habitat and starting to live their life, they certainly are going to be altering their habitat in some way. They're taking in nutrients, they're excreting wastes, they're breaking down things and recycling things. So lots of activities can cause that little habitat there to start to alter. And then that sets up the situation where secondary succession can occur, and so on and so on. And so you keep getting this process going until, if it's an undisturbed system, eventually you might get to a climax community, which is relatively stable, highly dependent on the habitat. And you can't have like big disturbances. Big disturbances would disrupt the climax community. So when you think about growth of microbes, this is looking at their reproductive rate. So that's, remember, that's birth minus death. So if they have a lot of births, so to speak, then they're going to have quite a nice upward curve as you plot it out. So generation is referring to their reproductive rate, or, you know, how many generations, every time E. coli reproduces, that's a generation. And so you just keep thinking about every time they reproduce, you have more and more generations. And then the population size on the y-axis. What they've plotted here is just reproductive rates that are slightly different from each other. 1.05 to 1.07 doesn't sound like that big of a difference. And yet you can see that when you plot it out over generations, you can get some very different looking curves. And what this curve is actually describing is exponential growth. When you see that swooping line like that upward, when it's plotted out in such a way where the X and Y axis are like this, that's a dead giveaway that you're looking at exponential growth, that they're growing faster and faster over time. You can also just do it the reverse way. They're dying exponentially. So this is a death curve where you can see that they're going to extinction with this data that they plotted out. But again, just a little bit of fussing with a reproductive rate can really impact their growth and their death in these different habitats. So going back to this idea of the carrying capacity in a habitat, that eventually, unless there's some big influx of nutrients or some way to take away waste, perhaps, eventually you can imagine where the carrying capacity of that habitat is going to be met, and the microbes can no longer keep up that really rapid growth. And they're constrained by resource limitation, maybe severe drought, or toxins in their environment. Their waste products are starting to build up and it's impacting them. And so when you work with microbes in the lab, it is very much you're thinking about this idea that there's going to be a carrying capacity, because the common way to grow up microbes in the lab is called batch cultures. So when we grow up any microbes that you used early on this semester, this is what we were doing. And when I do research with my Streptomyces bacteria, this is what I'm tracking. I'm tracking their growth in a batch culture system for the type of experiments that I need to do. And so this is the idea that you pop the microbe. So when we grew up E. coli for you, when you did the streak and spread plate and some of those other microbes, what we did was we took the microbe out of the deep freezer. They were frozen. And then we popped them into some broth and we put them on a shaking platform shaker that shakes that broth to help mix things and get the oxygen working through that broth so they can have access to oxygen. And what you'll see is that broth starts out very clear initially, it's crystal clear. And then over time, as the microbes are reproducing themselves, it gets cloudier and cloudier. Fancy word for that, by the way, is turbid. I think I might have mentioned that at some point, turbid. So in a micro lab, you, instead of saying cloudy, you tend to say turbid. And here you're tracking their growth over time. So on the x-axis is time, and on the y-axis you have optical density. So you take that broth and you take a sample and you put it into a spectrophotometer and you read their optical density. Oh, there's the word turbidity. And then you can track their growth. So this is very handy, just using a spectrophotometer, and you track the turbidity over time. And as you do that, that's the red line here where you can see, or no, it's the green line. Sorry, it's the green line that they're doing the optical density. Okay, so that green line. And you can see that pretty much any microbe that's grown in a batch culture like this. So batch culture means you pop them into the broth and you leave them alone. You don't add in any more nutrients. You don't take away any of their wastes. You just leave them alone and let them grow. And when you do that, you get this very classic curve. No matter what microbe you work with, it might stretch out. So for a slower growing microbe, this might really stretch out. But the same basic pattern is what you see here. So this so-called lag phase, if you take that and work down, so this area here is what they call the lag phase. So once I take E. coli out of the deep freeze and we pop it into broth for you, that's a big switch. It was frozen and dormant. And then you pop it into broth and it's like, okay, what's going on here? It wakes up and then it has to assess everything around it in terms of what kind of nutrients do I have. And then it has to get the gene expression up and running for those nutrients. And so you don't really see any growth here. It's kind of flat initially, a green line. So what that's telling you is there's no reproduction going on, but they're certainly busy because they're assessing their environment and figuring out what do I need to do in order to grow. And so they're still doing a lot of stuff. It's just not reproduction. Then they kick into reproduction, and that's when you get typically exponential growth. So that's what they call the exponential phase, where you see that nice straight line. So um, when you do the type of graphing where you have time on the x-axis and then you have the log. So you have a logarithmic scale on the y-axis. This is called a semi-log plot. A semi-log plot because it's only one axis that is logarithmic. The other one is arithmetic. So it's a semi-log. And so if you are tracking exponential growth and you plot it out in this way, it's going to give you this nice straight line that you see there in the exponential phase. So this is also exponential growth. It's just that they use an arithmetic scale on both the x-axis and the y-axis, and then you get a swooping line. So that's also exponential growth. It just depends how you plot it out, how it's going to look. Microbiologists like to do this. They like to plot out growth in this way with a semi-log plot because that gives exponential growth. You can plot that out. It gives you a nice straight line. Then you can take the slope of the line and use that to actually figure out population or how long it takes to reproduce. So getting a straight line can give you some good data off that. You can tell Excel to take the slope of it. And then it's easier to track visually. Very often when I'm running experiments, what I'm doing is I want it to get into, I want my streptomyces to get into exponential growth. And I'm going for antibiotic production and quorum sensing signals. And I know that they produce those during the second half of exponential growth. So I have to track that over time. And once I see them kicking into mid to late exponential, then I can start taking samples and testing it for the chemicals that I want. So exponential growth is considered, when microbes get into that phase, it's considered a very healthy phase for them because they have all their gene expression done, up and running. They're taking in nutrients, they're reproducing exponentially. So this is a very healthy point in their life. But if it's batch culture, that means you don't put in any more nutrients. So eventually the carrying capacity is going to get met. And that's what you're seeing with the stationary phase. You've reached carrying capacity. And so now their growth is leveled off because there's no more nutrients. And then when you get into the death phase, what's going on there is it's a exponential decrease because now not only is there no more nutrients, there's also toxins. Their own waste products tend to build up, and many of those can be toxic and they start lysing or breaking open. And so you can get an exponential death going on. So this is very, very classic when you grow microbes in this kind of batch culture system. This is explaining in words what we just went through. And so I would like you to be familiar with that graph and explaining a little bit about what's going on in each phase. Be able to maybe, I might ask you to like sketch it out and label it, that kind of thing. This is such a classic microbiology lab process. I mean, over and over and over, thousands of experiments have been published where they use this batch culture method. And so I would like you to be familiar with this concept. Now, there's plenty of other experiments that are done not in a batch culture. batch cultures are very easy because you don't, once you set up the experiment, you're done. You don't have to siphon anything off, add anything new. So it's a very straightforward experiment. And it can be totally adequate for certain questions that you might ask experimentally. Then I mentioned that chemostat idea. The chemostat is another very common method that microbial ecologists use because that's a fancier system. The equipment for that is, of course, way too expensive. But basically it has ways for you to put in sterile nutrients, usually some kind of broth recipe that you can add into the system at regular intervals, and then you're regularly taking away the toxins. And so you can keep basically a chemostat. Very often what we're going for is we want the microbes to stay in exponential. So once you get to know your microbe, you can put the chemostat, you can set the controls so that it puts in the broth, new broth at a very particular rate, gets rid of the toxins at a particular rate, and then you keep them at exponential. And then you can ask all kinds of questions. That's often done with, and they're looking at how do microbes behave with each other, if they're growing with each other. They'll tend to use a chemostat and look at their interactions when they're vigorously growing. So when you look at succession in a more realistic habitat, not in a broth culture, what you tend to see is that microbes that come in and the pioneers, they tend to be our strategists, where they can come in. It is a pretty uncompetitive environment, lots of resources, so they can just grow really quickly, take advantage of all of that nutrients that is pretty widely available to them. And so our strategists tend to have high growth rates as they come into a new habitat. And then it's also when there's not many other microbes around, so there's low population densities, they really can go and grow quite successfully. And then you can imagine as the carrying capacity starts to get closer, the case strategists will come in and be the more successful. And so again, through the successional processes, you can see how our strategists tend to be the early ones in setting up camp and growing very rapidly. And then the case strategists tend to persist in crowded habitats. So here's a graph showing two different populations, population 1 and population 2. And on the x-axis, you have a couple of things going. You have population density running off to the right, so increasing population going off to the right. And then substrate availability is very high at the left and goes lower as you work your way to the right. And then the y-axis is looking at population change. And then they plotted out the R versus K continuum. And so when they look at this and plot it out, they're just doing two different populations. But when you plot out this data, you can get a feel for which population seems to be more like an R strategist and which might be closer to a K strategist. So here, if you go to this area on the x-axis, so what that's showing is of low population density and high substrate availability. Does that make sense? So who would be better with low population but high substrate, an R or a K? Absolutely. And so the fact that R1 plotted up higher here in terms of the rate of population change. So population 1 was like cooking along really fast under these conditions of low population, high substrate. So relative to each other, population 1 is an R strategist. It seems to favor that, okay? And then R2 is just the opposite. R2 is more of a K strategist where it is okay, it can survive well up to the carrying capacity. And so these types of graphs are sometimes done when microbial ecologists are trying to look at these different populations. So that's how you kind of think and interpret that type of graph. So what we've been talking about with these successional processes are the idea that, erase, it won't let me erase. I don't know why it's doing that. Anyways, what we've been talking about is this autogenic succession. Autogenic. Auto means self. Genic means creating. Self-creating succession. Self-creating succession. So what that's saying is that you have those pioneers coming in, setting up camp, living their life, and they are modifying their habitat through just being there and living, doing their thing. And then once they are there for a while and they've altered the habitat, that allows other microbes that previously couldn't come in before to come on in, because now the habitat's more suitable for them. Then there's this other idea, allogenic. Okay, that's where the environment is selecting. All right, so the environment, the factors going on in the environment are what's causing any habitat alteration. So that a classic example is seasonal changes. So certainly around here with fall, winter, spring, and summer, we definitely have pretty extreme seasonal changes, and that's helping this allogenic succession idea. So they can track this type of process and kind of pinpoint, is it autogenic or allogenic, depending on the ecosystems or habitats they're looking at. This is in a mouse, I believe. Succession of microbial populations in the large intestine of mice following birth. So this is a baby mouse, and they were looking, the way they track this is through fecal samples. They would take fecal samples and assess the microbial population over time. So you have this, the x-axis is on days, and then the y-axis is looking at the log growth of the different bacteria. So they have 1, 2, 3, 4, 5 different groups of bacteria listed here. And I think it's really obvious as you scan top to bottom or bottom up, and you look at these different graphs, they're not all identical. And so you definitely have some successional processes going on here. So it looks like there's a couple of ideas here. So the pioneers look like Flavobacterium came in early on. That was popping up early. But so was Lactobacillus and Streptococcus group, as well as the Enterococci. They were a little bit low initially compared to the other two, but they look like they were, pioneers coming in into the intestinal tract. Could have got it from the mom or the environment. The baby was getting that introduction. And you can see that they were persisting, but it looks like Flavobacterium couldn't cut it, right? All of a sudden you see its population cut. So the habitat for Flavobacterium was unsuitable for some reason. Maybe it got outcompeted. Maybe the environment of the intestinal tract was altered in such a way that it just couldn't sustain living there. And so that one phased out. But you can see that the other two up top, they came in early and they persisted, and they did quite a good job here throughout the experiment. But then you see like Bacteroides not showing up till much later. So again, that's implying that Bacteroides is not a pioneer. It needed to wait for the habitat to change, conditions to change in order for it to get in and colonize. So when you plot out data like this, you can get a little bit of a feel for the successional processes going on. In this case, it's in the intestinal tract.
Possibly a silly question, the space that goes through all the way down on the right side, is that just like a space in like when they were?
Yeah, I think they didn't plot or didn't take samples in this, they waited. Probably due to expense, could be my guess. Because in a perfect world, you keep going all the way out to 20 days. But I think what they did was, well, we have enough funding to do it early on, and then we'll wait a little while and do one last sampling and see what we get. So this is a good example of autogenic, right? The gastrointestinal tract is fairly stable environment. And so most of the successional processes here are self-generated by the microbes themselves, living there, altering the habitat, letting others come in due to that activity. And then here you're looking at seasonal succession going on. This is a very long timeline from 1964 to 1967 instead of days. And then relative abundance of different diatoms in a thermal spring of all things. And again, as you work your way through these different graphs, you can definitely see that there are some differences, but there's this rhythm to the seasonal changes going on. That you can also see this kind of pattern in each one. There's this kind of ebb and flow going on with the seasonal changes. And then as you compare each of the different ones, the graphs are slightly different. Some are more modest abundance and others are quite nice. So, but again, this is a allogenic idea where the microbes have no control on the seasons. It's just, that's what their environment is giving them and they're dealing with it, but that's what's causing these successional processes. Severe disturbance are often something that the question that the microbial ecologists are asking, like after a severe disturbance, what happens to the microbes? Very similar to like when Dr. Robertus does a lot of his research, he looks at severe disturbances, whether it's beaver activity coming into an area, you know, where they weren't there before, wildfire sweeping through a forest system. He tends to look at that kind of idea where he's looking at severe disturbance and then how does the ecosystem rebound from that? And very, very interesting research. You can do that at the microbial ecology level too, where you're looking at severe disturbances. In this case, they're looking at, they had volunteers not wash their hair for over a week, basically. And so they took samples of the scalp and determined who was there. So this line running through all the graphs, that's the before wash. So they were taking samples and you could see that they had all of the different microbes were there before the wash. And then they had them wash their hair. And you can see that after they wash their hair and sampled again, it looks like, for example, in the bottom, the criniforms got hit where they didn't persist after the wash too well. They were there, but they got knocked down. And you can see the, they just call them the aerobes. They got severely hit, but not so much with the Propionibacterium acnes. That one was like, I don't care that you washed. I'm fine. I can persist and live through that. And actually it seemed to stimulate their growth because maybe they had less competition. And so then they gave a brief flourish after the wash because they were all of a sudden, maybe they were devoid of competitors around them. And then you can see, okay, now don't wash anymore for the next week. And what happens? So, you know, washing your hair is a severe disturbance for microbes on your scalp for sure. And so you can see that the microbes in general started to come back, right? You start tracking all the way out as they took samples over the days. They kept going pretty much up and up. But then look at day 7. Look at all those bar graphs and look at before wash. What kind of conclusion would you say about those the comparison between before wash and day 7. Yeah, they're about the same. And so basically, the conclusions they were coming to from this study is that, yes, washing your hair is a severe disturbance. However, given enough time, leaving it undisturbed, the population reverts back to what it was. And so this idea of severe disturbance, in this case, it had a modest effect, but the population was able to reestablish and stabilize back to what it was. So this was not exactly what they were hypothesizing. They thought it wouldn't be so neatly comparable like this. So that was kind of an interesting study. One of my former grad students, Andrew, he did dreadlocks for a while, and I think he chose not to wash his hair. And I just kicked myself that I did not we didn't do the sampling with him. That would have been a really interesting experiment to see. And he would have been totally into it. But he doesn't have dreadlocks anymore. I just saw him the other week when he came back to visit, and no dreadlocks. Okay, so this idea that back to the original community, after the disturbance, they were able to reestablish back. Now, is that always the case? No, that's not always the case. It's just this particular study, you were able to see that original community pretty much coming back. Now, you can also apply this, maybe not just to scalp, but thinking about the human body. This is data taken from a published paper where they were plotting out bacterial diversity and populations after antibiotic ingestion by the patient. So day 0 is before the patient went on antibiotic therapy. And the different colors on the bar graph are different microbes. So pretty good diversity. Then they started taking antibiotics. How long does your doctor typically tell you to take antibiotics generally? 3 days. I've seen 3 days, 7 days, 10 days, roughly. So 7 days, so after 7 days of treatment, and then you stop. Let's say this patient stopped after 7 days. Well, the day 7 graph, it's all green. There is no diversity. There's like one group of microbes is it. So then they stop taking antibiotics after day 7. Look at day 21 days out. There's no rebound. 3 months out, no rebound. 6, 9. That's rather alarming. 9 months out after antibiotics. He stopped taking antibiotics. 9 months out. Still, now we're close to what he had beforehand. Then a year later, you finally start to see some other colors popping out besides green, which means you're getting more diverse microbes showing up. But let's look at the last reported data, 24 months, 2 years. Compare 24 months to 0 days. Are they the same? No. When this was published, scared me. It scared me for all the people, including myself, who have taken antibiotics. And I'm pretty sure probably most of us in this room have taken antibiotics, either prescribed by a doctor or, certain things that you might ingest have antimicrobial properties as well. So anyways, now this was a very small sample size. So let me just say that this was only, I think, the number of patients they tested was like a sample size of, it was 5 or less. So you got to take that with a huge grain of salt. But there have been other studies that have come out since this one. This was one of the earlier studies published that made people go, uh-oh. And then they started looking at this more carefully. And now with better technology of being able to take it's fecal samples. You can't exactly go into the human intestinal tract and sample, although they're working on doing that, but they haven't succeeded on doing that without an operation that would not work for the experiments. So it's fecal samples that they're testing. So that's also a limitation. Although they're working on, is it Cornell maybe, is working on the idea that you swallow a pill and it actually has like, it will actually take samples, like take little snippets as it's making its way through your gastrointestinal tract. And then you poop it out and they grab it and they can, I don't know exactly how they were doing that, but it's in the works. They're having success with pigs. They've had a pig swallow it and had it go through, and they're making progress on it. So once they get something like that established, oh boy, is that going to be they're going to be rich because all the researchers are going to want something like that. Because right now you can't, when you take a fecal sample, you don't know, well, did the microbes in these species, are they primarily from the small and, coming from the small intestine, large intestine, where are they? And with that ability to sample along the way, you could get at that question. So, okay. Also, don't forget that when you look at communities, it's not all about competition amongst themselves. You have cooperation going on, and that's often what keeps that population stable, is there's a lot of cooperation going on, and therefore the community stays stable. And yes, there's competition too, no doubt. But often I hear a lot about the competition and not enough about the cooperation. And well, to be fair, I guess decades ago, they just assumed microbes were always trying to kill each other and compete, and they didn't understand that there was cooperation going on. But now we know better. So this idea that you certainly have cooperation, you have competition, certainly a lot goes into impacting that. How are we going to coexist? You have to deal with allogenic processes. You have to think about your resources, how patchy or heterogeneous, spatial heterogeneity is a fancy word to say how patchy is your environment. And so thinking about that, where there might be an area of the habitat very devoid of nutrients, and then right next to it, really flush with nutrients, for example. Then you got all kinds of predators to worry about, whether or not they're going into dormancy or long-term dormancy. What about severe disturbances? So all of this you need to think about. All of that's going to influence both competition and cooperation, really. And so it can get very, very messy. And then don't forget about genetic exchange going on. You have that kind of interchange amongst the individuals of that community. Speaking of which, so here's some data with genetic exchange going on and mutation influences. So 1980 to 1990, so a decade. This is antibiotic resistance, penicillin resistance to the pathogen Neisseria gonorrhoeae, causes gonorrhea. It's used to be very sensitive to penicillin, but that's not what this graph is showing. So a decade of, back in 1980, virtually no resistant pathogens. Just a decade later, look at the amount of resistant microbes. So what this is showing is that when you started putting penicillin out into the population, the human population, and then, so that's a selective pressure right there. When you start introducing antibiotics, that's a selective pressure on the microbe. It's allogenic in that regard. And so those microbes that could handle the presence of penicillin persisted, pass that trait on to their offspring, their population grows, the sensitive ones start to crash, and pretty soon you get this shift of antibiotic resistant microbes really dominating. You can show that over and over. There's all kinds of published studies showing antibiotic use here on the x-axis, and then percentage resistant on the y-axis from fecal samples. And you can see many different antibiotics here. So tetracycline has been heavily used, given to humans for a variety of different illnesses. And so that one has been out in the common use for many, many times. And you can see the use, the tonnage usage. Tetracycline's been used a lot. And so that one has the highest resistance. The ones that aren't used very much, gentamicin, only, under 10 tons of it be out in the environment. And you can see not much resistance. So this is a very nice graph showing selective pressure. You have plenty of tetracycline, you're going to get plenty of tetracycline resistant microbes. Take that away, and eventually it'll drop. Microbes are very good at purging. If it's no longer effective for them to maintain that gene, they'll purge it. And so you can alter this. You could change tetracycline to be much lower just by removing tetracycline from the human usage, because then you're removing that selective pressure. Here on the on the figure B, you have again the Neisseria idea where they tracked it over time, and you can see the resistance as penicillin continues to get used, you get more and more resistance. So that's a problem. Antibiotic resistance, I think most of you are aware of, unless you have lived in a cave up to this point. It's kind of common knowledge that antibiotic resistance is a thing. This is just showing some that are resistant. Different types, gram-negative, gram-positive, fungi. And this graph is showing antibiotic resistance. So you can see Staph aureus was kind of the first to start gaining resistance. So I can't help myself. I got to explain this idea, because when you think about antibiotics as a selective pressure on microbial ecosystems, habitats, so in your gastrointestinal tract, let's say this is happening. You go to the doctor, you're sick, he gives you an antibiotic, you start taking it. Oops. So on day 0, when you go in and you're sick, if you mapped out your microbes, you would find that you would have, well, let's say it's salmonella. Let's say you're sick with salmonella in your gastrointestinal tract and it's causing food poisoning. Start taking antibiotics. So salmonella, some of the individuals with salmonella are going to be highly sensitive to that antibiotic. Other individuals of salmonella will be modestly resistant just by their mutation, their natural mutation that goes on. Some are going to be highly resistant. That's just the nature of it. Some individual cells have different sensitivities to the antibiotic. So you go to the doctor, he says, take these antibiotics. So you do. So by day 3, this idea is going on inside of you. You have the really sensitive ones have started to crash. The intermediately sensitive ones, you're knocking them down, but your resistant ones, they haven't really changed. So you've altered the population through antibiotic use, but not really to your favor yet. Day 6, now you see the really sensitive ones are gone. Most of the intermediate ones are gone. What about the highly resistant ones? Are they gone? No. So can you imagine if you're a highly resistant organism living inside you? What is day 6 depicting? Would you be happy if you were one of those highly resistant organisms? Living in your gastrointestinal tract, you're highly resistant, your populations are still high, your competitors are virtually non-existent. That's like party time. That's a good situation. If you were highly resistant, you'd be like, whew, thank you, pills. You've wiped out my competition. I'm good to go. Day 6 is usually when you start to feel better. And if you don't have a lot of money, you don't want to go back to the doctor again, you have some pills left, what do? You save them. You don't keep taking them. You put them in your medicine cabinet and say, I'm going to save these for next time I get sick, so I don't have to go to the doctor. That's what they're showing in this shift here. If antibiotics are stopped prematurely, you just set the stage to let the resistant microbes in you win. So now you stop taking antibiotics, they're still there, and they just explode in their growth. Now you get more sick with resistant microbes. Not only that, let's say you step into a elevator or whatever, and you start coughing or hacking or whatever. If you can transmit this to other people, now you're giving other people highly resistant microbes. So it's not just a you problem, it's a community problem. If you continue to take those drugs, what happens is it's not so much the drug, you're like, well, they're highly resistant, no matter how much you take it, it's not gonna work. But what can happen here is some of the highly resistant cells could mutate just randomly to be sensitive. So you could knock them down still. The other idea here is that you're keeping any sensitive ones knocked down, and then your immune system typically has, by then, everything up and running where they can finish off any lingering pathogens. So between random mutations that can cause them to become sensitive to the antibiotic, plus your immune system, it tends to do the job of finishing off the pathogen. But if you stop early, you're just creating a perfect storm. So now that you have a little bit of microbial ecology background, I'm hoping this thought process makes more sense. And I really urge you to talk about this to other people, because most other people don't have this training and they don't understand this. And like I said, it's not just a you problem, it's a community problem. If people keep walking around with highly resistant organisms, that's a really good way to get that spread out into the population. Make sense? Okay. All right. Let's stop there. And we're going to get into habitats next. We'll start talking in a very big picture way about microbial habitats. We'll start that on Monday.